Hello everyone, this lecture video is all about hemoglobin catabolism. And red blood cells are highly specialized cells whose primary function is to deliver oxygen to and remove carbon dioxide from body tissues. The mature red blood cells have no nucleus or DNA, so instead of processing the oxygen there, they are filled with red pigment hemoglobin to aid the delivery of the oxygen. The red blood cell formation occurs in bone marrow, and about 200 billion new red blood cells are formed daily. The lifespan of red blood cell is about 4 months, and this oxygen-carrying ability of red blood cell is due to the protein hemoglobin present in it. Hemoglobin is a conjugated protein. The protein portion is what we call the globin, and the non-protein or prosthetic group, when we say prosthetic, that is being attached to the protein portion, is the heme. That's why this is called as hemoglobin. Heme contains 4 pyrrole groups joined together with an iron atom in the center. This is the structure of the heme. So this is your iron, and this is the pyrrole group. We call this as tetrapyrrole because they are 4. And it is the iron atom in the heme that interacts with the oxygen gas, this part. So this one is positive, right? And the oxygen is negative, so that is why there is an attraction between them. And then synthesis and degradation of hemoglobin. The old red blood cells are broken down in spleen and liver. The globin protein is hydrolyzed to amino acids, which become part of the amino acid pool. The iron atom in heme becomes part of the ferritin. The ferritin is an iron storage protein which saves the iron for use in the biosynthesis of new hemoglobin molecules. Then, the tetrapyrrole carbon arrangement of heme is degraded to bile pigments that are eliminated in feces and to a lesser extent in urine as well. The degradation of heme starts or begins with the ring opening reaction in which the single carbon atom is removed, and the product is called biliverdin. So this is the heme. So the ring here will be open when it reacts with the molecular oxygen gas. So when that happens, the 3 important components of this water molecule will be released. The iron here will be released as well as ferritin. Okay, that will be used again in the production and synthesis of new hemoglobin molecules. Then we will consume here the reduced form of nicotinamide dinucleotide phosphate, and that will be converted to its oxidized form. So when that happens, the heme will now be a biliverdin. This is the new form. So the second step of heme degradation, biliverdin here will be converted to bilirubin, and this change involves reaction of central methylene bridge of biliverdin. So this M here is methyl, this is a vinyl group, the letter V, and the P is the propionate. So this part here, 1, 2, 3. A sugar will attach on this part to make this one soluble in water. So the sugar is this one that will attach on that part. When that happens, the biliverdin and bilirubin will have a more solubility in water and making it more easily to excrete in the body. Then, an important discovery in 2002 occurred. Bilirubin has an antioxidant properties according to the research, and it protects against peroxyl radicals which are toxic in our body by being oxidized back to biliverdin. Its antioxidant properties are significantly better than those of glutathione, which for 80 years believed to be the most important antioxidant in our body. This bilirubin is found in low concentration in cells but in higher concentrations in blood. Then the research suggested, based on these findings, that bilirubin is the major antioxidant protector of cell membrane, while the glutathione protects the inside part of the cell. Now, when the bilirubin here has been attached to a sugar, it makes it more soluble in water. What will happen is the conversion of bilirubin into parts in urobilin and stercobilin. So, this urobilin will be excreted in the form of urine, and stercobilin will be excreted in the form of feces. Next, we have here bile pigments, the tetrapyrrole, the 4 nitrogen attached on the iron earlier, this one. This 4 pyrroles here or tetrapyrrole will be removed in the body. The tetrapyrrole degradation products obtained from heme are known as bile pigments. So when they had been excreted in the process and removed in the attachment, they will be known as bile pigments because they are secreted with the bile, and most of them are highly colored. So we can define bile pigments as colored tetrapyrrole degradation product present in bile. And then biliverdin is green in color from the word verdin, it means green, and bile here, it means bile. So bilirubin is reddish orange in color. Rubin means reddish or red or orange in color in Latin, and bile means bile. Stercobilin, the cobalt part here, it means brownish in color, and the compound that gives feces their characteristic color, the brown color of the feces. And then urobilin here is the pigment that gives urine its characteristic yellow color. From the euro, it means urine, it means yellow. Okay, so that is how they are being excreted. Biliverdin and bilirubin is also being excreted in the body in the form of feces. But then the concentration of stercobilin is higher compared to the two. So that is why the color of your feces is sometimes brown. But then there are instances that the color of your feces is color green. Meaning to say the biliverdin there is the concentration of biliverdin is high, or sometimes it has like a blood color in your feces. That is because the bilirubin concentration is high during that time. So next, normally the body excretes 1 to 2 milligrams of bile pigments in urine daily, and 250 to 350 milligrams of bile pigment in feces daily as well. And then what else? When the body is functioning properly, the degradation of heme in the spleen to bilirubin and the removal of bilirubin from the blood by the liver balance each other. So, meaning to say, if they are balanced, their colors wouldn't manifest in the feces and in your color as well. But if there is an imbalance in this part, we will have the disease called the jaundice. It occurs when there is an imbalance, making the bilirubin concentration higher than normal. So when this happens, the color of your feces has a tint of red, and the skin and whites of your eye acquires a yellowish tint. So from the French word jaune, it means yellow. And the causes of jaundice is first liver disease such as cirrhosis, when the liver is malfunctioning due to the accumulation of fats, and spleen malfunction and gallbladder malfunction. A mild form of jaundice in premature infants occur as well because of their underdeveloped liver. So that's why they are sometimes in the generally colored yellow. So the treatment for that is the doctor will use white or UV light, which breaks the bilirubin down to simpler compounds that are more easily excreted. Then, the local coloration associated with deep bruise is also related to the pigmentation associated with heme, biliverdin, and bilirubin. The changing color of the bruise as it heals reflects the dominant degradation product present in the tissue repairs itself. So, sometimes it is color red. The present during that time is bilirubin that is being absorbed in the tissue. And sometimes, in the long run, it will turn into brown due to the stercobilin, the color dominant during those time. So, that is all for hemoglobin catabolism.